from the important fisheries resource in the Europe, and then the marine mammal and the top predators. And having that said, combining this effort in understanding and the top predators would enable us to promote what kind of mammals and the top predators. So having that said, I hope we have a interesting what kind question from the top predators. So having that said, I would like to invite maybe interesting question from the top predators. The question I would like to invite and maybe interesting question from the top predators. The question I am going to invite and maybe interesting participation from the top predators. The question I am raise hand and maybe interesting participation from the top predators. The question I am raise. Deliver your question in Bahasa, and I can freely. Okay, we have Pak Charles, and maybe there is a second participant who would like to ask to our distinguished speaker this morning.
I cannot find the raise hand tools here, so maybe I can ask for another second.
I see we have two participants who raised hand, and I see Pak Charles and Pak Fedi Sondita. Okay, the floor is yours, Pak Charles. I see we have and then Pak Fedi. Participants who raised hand, and I see Pak Charles and Pak Fedi Sondita. Okay, the floor is yours, Pak Charles. Okay, and then Pak Fedi. Pak Charles, your voice is very small.
Can you hear my voice now?
Yes, it's much more clearer. Please continue.
Okay, thank you very much, Dr. Andriani, and thank you for three invited speakers. Thank you very much, Prof. William. Thank you very much, Prof. Reggie Sensei, and also Dr. Paulo. My question to Prof. William as well also to Prof. Reggie. To Prof. William, my question, thank you very much for your excellent presentation. In your presentation, you said that small pelagic fish such as sardines have a dense of micronutrient species, but the level of fishing vulnerability and also vulnerability to the climate change is high. Since in Indonesia we have so many species of small pelagic fishes, so could you please emphasize, what kind of how we can manage our small scale fisheries that targeted species is the small pelagic fish. That's my first, due to the resilience to the climate change. And my second, and my question to Prof. Reggie Sensei, thank you, Sensei, for your fascinating study where tropical fishes thrive in Japan, especially locations that receive heat discharges from the nuclear power plants. Regarding the tropical fishes that found in Japan, do they originate from fish larvae that origin from the tropical region that drifting by the Tsushima current or Kuroshio current, or the origin from the locally spawned fish larvae that have already adapted well to the Japanese water? So that's my question. Thank you very much, Dr. Andrian.
Okay, thank you. We are going to request Pak Fedi to deliver his question first. No, not the first, the second actually. Please, Pak Fedi.
Thank you, Dr. Sanudin, Adriani Sanudin. Okay, thank you to Professor William Chong. Okay, I noticed that the discussion on your presentation mostly on the pelagic species, commercial pelagic species, probably because that most likely affected by the impact of climate change. I want to know how much studies have been done is the world on the demersal fish and less mobile commercial fish or animals, yeah, due to changes in the oxygen and acidification. And to Professor Reji Masuda, I just wonder, what is your suggestions if you want to make a long-term monitoring for species that heavily exploited by commercial enterprise? And to Professor Paulo Momigliano, thank you for presenting the beauty of the genetic studies, but I want to know, probably you have some knowledge on what is the gene that help organism in marine organism that help them to have a weight tolerance on facing the changing environment. Thank you very much.
Okay, thank you, Pak Fedi, for the question to all the three panelists. And we have final question coming from Andrianus Sembiri. Please, Pak Andrianus, mention your organization. Also, yeah, don't forget to open your camera. Thank you.
Yeah, thank you, Ibu. My name is Andre from Bionesia. My question is to Paulo. You said in your first study case that you found there's a pattern on the genetic population. My question is, what type of marker do you use to study the population genetic in your first study case? And do you also found that like climate change or a different climate that also affect the result or not? And the second question is, you said in the last your presentation, you said that focus on the pattern, not only focus on the process, not only on the pattern. What's that mean? And based on your research on the shark genetic population, especially in Indonesia, what is your recommendation for Indonesian shark fisheries management perspective based on what you found in your study, especially for Indonesia? Thank you.
Thank you very much. So I think we have all three panelists have their own share of questions. To William, I think you have two questions from Pak Charles, your fellow colleague here in Indonesia in doing the Sardinella research in Bali. And also from Pak Fedi, both are requesting suggestions on how to manage small-scale fisheries and also demersal fish as impacted due to acidification or deoxygenation. The second one is to Professor Masuda. Both questions come from Pak Charles and also Pak Fedi related with biomonitoring suggestion on fish larvae and also for the heavily exploited demersal fish. The last panelist, Professor Paulo, received questions, some technical, and I think it will be very beneficial for us here in Indonesia to mainstream the genetic or genomic research for future biodiversity conservation. Please, the floor is yours from Professor William Cheung, then continue to the second panelist, Professor Masuda, and finally, we will have the answers from Professor Paulo.
So thanks for the questions about the small pelagic fisheries. I think to help more specifically small pelagic fisheries to adapt to climate change, I think there are a number of ways. I think some of that I mentioned in my talk, but more specifically for small pelagic, I think there are two aspects. First of all, is to understand the current is to reduce the or improve fisheries management, sustainable fisheries management on the fisheries, because we know the small pelagic fisheries are also prone to collapse. And we know it from other examples, for example, in other systems where when overfishing combined with the environmental change impacts, it could actually drive collapse of the small pelagic fisheries too. So what it means is that it's really important to manage the fisheries in a way that can maintain also sufficient biomass, and not only that, it needs the sustainable biomass is needed to support the human fisheries, but that because small pelagics is a forage fish that is also important for other ecosystems as well, and often the changes in the ecosystems are associated with overfishing of small pelagic fish, for example, in addition to climate change, can drive shift in ecosystem structure. And so I think in this case, it is also really important to manage fisheries so that we can maintain high level of biomass. And then more technically, I think small pelagic fisheries are fairly sensitive to short-term environmental change, particularly, for example, things like climate heat waves or other extremes or compound events associated with the long-term climate change. And with that, I think more scientific knowledge about short-term predictions of what changes, what driving changes in small pelagics, abundance, distributions, and also the ability to detect and predict their changes would be really useful to support fisheries in adapting and developing management strategies to manage the shifting biomass associated with these more short-term extreme events, compound events impact. So is that I think those are the two things that are more specific to small pelagic fisheries.
Okay. Thank you, William. I think we got the message very clear. And the second one, please, Professor Matsuda.
Yes. First, I was very impressed with Dr. Chen's presentation, and especially I totally agree with his conclusion that we should efficiently utilize small pelagic fishes. And I actually love small pelagic fish such as anchovies and sardines. And simple solution is just utilize and just eat them by a different way. I often cook like anchovy both by fresh or by Italian cooking, and they are very nice to eat. So rather than feeding them to animals or larger fishes, just eat by ourselves. That's a simple answer to utilize them. And for the question of recruitment of tropical fishes in our waters, basically they are from tropical Japanese waters such as Okinawa through Tsushima warm current. There evidence is that we found them only in only in autumn season, but recently we find more individual in late summer. So the spawning area is becoming more and more northern area. So they may start to self-recruitment in our waters. For the third question concerning monitoring of commercially heavily exploited species, underwater, there's a limitation of power in underwater observation, and my talk today is focused only on underwater observation, but in some cases, analysis of catch may be more efficient to get data. And recently, or actually more about from 10 years ago, we are conducting analysis of environmental DNA, which is collecting just water and analyzing what species should be in the water. And that's sometimes very powerful and provides lots of valuable information. So we are conducting monitoring of both underwater observation and also environmental DNA. And with such a monitoring and monitoring of water is can be conducted in parallel in several different places. So that's unlike underwater visual sensors. So we may try different way of monitoring. So that's my answer for those 3 questions. Thank you.
Thank you very much, Professor Masuda. Last one, please, Professor Paulo.
Genes are, I guess, involved in tolerance to future change. I'm not sure if I understood the question exactly, whether it was referred to like one of the specific example, but the short question is that, I mean, evolution is not so predictable, so we don't know. I mean, apart from some, you know, basic observation that probably each of proteins may be important, at least in the short term, in terms of managing the worst of heat waves in the marine environment. Apart from that, we don't really know what genes are going to be useful. So when I think about preserving evolutionary potential, I'm not thinking of preserving specific genes. I'm thinking about maximizing the variation in a species genetics. So basically, we know that the potential of species to adapt depends on their standing genetic variation. So the amount of the differences that there is in their genetic composition. So a species that has very few alleles, that is homozygous across the entire genome, is not going to be able to, doesn't have a very high evolutionary potential, because genetic differences can translate in phenotypic differences. The lower the genetic diversity you have, the lower the possibility for selection to act. So I hope that answered a little bit the question. So I don't think it's about, you know, trying to predict what's important, and I mean, there are things that we can do. For example, we might you know, decide that it's a good idea to study corals in areas that experience very, very high temperature, and they cope with it very, very well. Like, I'm thinking about places like the Persian Gulf, for example, because there may be, you know, there may be genetic variation that may be important for potentially other populations to adapt in the future. So in that sense, we can try maybe to protect some adaptation. But I think in general, we should try to protect genetic diversity as a whole. So try to make sure that we protect the species, the populations that contribute the most. And that means try to protect the populations that are the highest diverse, more they harbor the high diversity, but they also are more differentiated between each other. So they harbor more complementary diversity. Think about it a little bit as in the same sense as preserving both alpha diversity in ecology and beta diversity in ecology. The other question that I had was there were 2 questions from Andrianos. And the first one was about markers.
Yep.
Yeah. So for the first study, well, the first part was we used RAD data. So basically, we sample a very small portion of the genome randomly. It's about, I think, 1% of the genome, roughly, and sequenced it. There are many different kinds of RAD. Prediction, basically, the idea is that you take the genome, you use a restriction enzyme that cuts it in many places, and then you make random cuts around these sites, and you ligate some adapters, and you sequence this very small proportion of the genome, which is still millions of bases, so it's still a lot. For the first part we used that. For the second part of the first study, where I showed the selective sweeps of this area of under selection in the genome, for that we use whole genome data, which is becoming increasingly cheap, is competitive with doing rat data. The only downside is that the analysis takes much more time. It takes more space to store it, etc. So I think that unless if you're looking at selection and adaptation, then I think that you need to think about using whole genome data today. But if you're interested in understanding evolutionary history, if you're interested in understanding, genetic patterns, if you're interested into reconstructing demographic history, or for all the rest of the SHARC project, we used rat data, and that's totally fine. The reason why we have to use whole genome data when we look for selection is that finding genes under selection it's like finding a needle in a haystack. You can't just pick up a few you can't just pick up a handful and find it. You need to look everywhere, and you need to look in the whole genome, because you don't know a priori where selection is going to act. So basically, I think that I hope that helps with the question. And when I talk about processes, the other question was what I mean when I say that we don't need to look for patterns, but we need to look for processes. Is that as I was, if you see the example of the sharks before, I started by showing a PCA, okay? So a principal component analysis of genetic variation in a bunch of sample population. Now that tells us about patterns of genetic diversity, pattern of genetic differentiation. Okay? It tells me that the population that I sample are more similar and some are less similar, right? But it doesn't tell me how that pattern came about. It doesn't tell me what are the processes that generated that pattern. So if I want to come up with predicted model, for example, of genetic differentiation, I need to understand the processes that created that pattern. And in this case, we know that the process that created the pattern is a range expansion, and then after that, migration across a heterogeneous landscape, right? And this information that can help us better in management. So we can focus on what parts are better to protect. In terms of Indonesia, well, I mean, what we showed is that if you want to see there are different. I mean, this is going to be different from different species, right? But at least for the reef shark, we have we predict the differentiation between different regions of Indonesia, right? But so I mean, there's going to be different areas that will need to be managed differently. The issue is, right now we looked at one species. I know that Andrian is looking at some other species, but to really come up with some advice in management, we really need to start looking at multiple species. We need to identify the processes that determine the determinants of genetic differentiation and diversity across multiple species, so we can come up with conservation strategies that are not just for the prairie shark, or the silky shark, or the I don't know, whatever guitarfish. We really need to think about multi-species studies that are that unfortunately are lacking. I think that's where we need to go.
Great, thanks, Paulo.
Okay, thank you very much, Paulo. And to all our distinguished plenary speakers, we would like to thank you very much for your contribution, not just delivering talks, but the insights that you've gave to our Indonesian fellow particularly, and also our international panel that is attending the 2-day session of Embrio International Symposium this year has been very fruitful to develop further research ideas, or maybe organize further collaboration between parties. Because if we see from the participant that delivering the questions, we can see there is a need for us to collaborate further. Pak Charles is a larval ecologist, fish larval ecologist, and he have certain necks of climate change, and also studying social ecological system that may be promoting further fish conservation. While Pak Fedi is a conservationist, marine conservationist, and he prone to work with community, developing their capacity. While the other participant who delivered his question, Pak Agustinus, is we can say that his company is pioneer in utilization of biodiversity sustainably, and how this particular biodiversity can be developed into different kind of resources which can be beneficial. However, he would like to have further insights on how to promote this biodiversity research and also action collaboratively by utilization of science, and conduct this particular science to help future decision making, and in terms of decision in the future, so we can start mitigate the climate change impact affecting our